Hello friends, welcome to the discussion of the NEET 2019 biochemistry section. In this section we will be looking at the questions which were there in your January 2019 NEET exam. So without much ado, let's start the discussion on biochemistry. So first question in your NEET exam was on Zellweger syndrome. Zellweger syndrome is due to defect in which organelle? So let us quickly take a look at Zellweger syndrome in very brief. The Zellweger syndrome, also known as the cerebro-hepatorenal syndrome. These are the three systems which are involved. The cerebro-hepatorenal syndrome. First thing, the three systems which are involved are the brain, the liver and the kidney, so we call it the cerebro-hepatorenal syndrome. Second part about the Zellweger syndrome, defective organelle is the peroxisome. The defective organelle is the peroxisome. There are two possible defects. Either the peroxisome is absent, the peroxisome is not there. Either it is absent or it is empty. Meaning the structure of peroxisome is intact, but the enzymes which should be there inside the peroxisome, those enzymes are missing. So either it is absent or it is empty. Now peroxisome is very important for metabolism of the very long chain fatty acid. The small chain, medium chain and the long chain fatty acid get metabolized in the mitochondria by the beta oxidation. But the very long chain fatty acid do not go to the mitochondria, they rather go to the peroxisome and they get metabolized via beta oxidation like reaction. They get metabolized with help of beta oxidation like reaction. So when the peroxisome is not there, the breakdown of the very long chain fatty acid will not occur and results in the accumulation. It results in the accumulation of VLCFA. So two things to keep in mind. First, the Zellweger syndrome is due to defect in the peroxisome. Second, the predominant finding is the accumulation of very long chain fatty acid. Now please remember that very long chain fatty acid is mostly present in CNS, in significant amounts it is present in the CNS. So accumulation also occurs mostly in the CNS. Accumulation also occurs mostly in the CNS. Another point to keep in mind is the peroxisome is also responsible for metabolism of the branched fatty acid. It is responsible for metabolism of branched fatty acid. Typically in food we have what is known as a phytanic acid. However, this amount is small and therefore small amount of phytanic acid is also there in the background of a large amount of the very long chain fatty acid. So remember, in Zellweger syndrome the VLCFA is the predominant accumulating substance, but in addition the branch chain fatty acid also accumulates in small amount. In fact, there is a variant, the milder form of the Zellweger syndrome, where we only have the deficiency of the enzyme related to branch chain fatty acid, which is known as the Refsum disease. In Refsum disease, only the enzyme related to the branch chain fatty acid is missing and results in accumulation of phytanic acid, whereas in Zellweger syndrome all the enzymes of the peroxisome are either absent or the peroxisome itself is not there. So in that case the predominant accumulant is the VLCFA, whereas in the Refsum disease we mainly get the branch chain fatty acid accumulation. So coming back to the question, the answer would be peroxisome. The Zellweger syndrome is due to defect in the peroxisome. Moving on to question number 2, which of the following will be elevated after a fatty meal in lipoprotein lipase deficiency. Lipoprotein lipase is responsible for breaking down the triglycerol. The lipoprotein lipase will break the triglycerol in two of the lipoproteins. Which of the two? It will break the triglycerol in chylomicron. It will break the triglycerol in VLDL. Luckily, VLDL is not there in the option, so there is no confusion that if LPL is not there, whatever chylomicron is formed after the fatty meal, it cannot be reduced in size, it cannot be converted to the chylomicron remnant. So the chylomicron will stay at elevated amount after the meal. Once we have the meal, all the lipids are secreted in the form of chylomicron. Chylomicron contains the exogenous triglycerol, which has to be metabolized via the lipoprotein lipase. So if lipoprotein lipase is not there, this exogenous triglycerol cannot be metabolized, and therefore the chylomicron will remain in the circulation for a very long time. So the lipoprotein lipase deficiency will present with elevated chylomicron level. This is part of the primary hyperlipoproteinemias, where one or more genetic defect can be there. In fact, we have the 5 main types of the primary hyperlipoproteinemias, and you can see the lipoprotein lipase deficiency is the type 1. It is the type 1 primary hyperlipoproteinemia, where the chylomicron is elevated. Coming to type 2A, which is again very frequently asked. Here you can see the defect is in the LDL receptor. LDL receptor is deficient, resulting in accumulation of LDL. This is very, very important because it is very commonly asked, since LDL, as you very well know, is associated with atherosclerosis. As a result, among all the hyperlipoproteinemias, the strongest association with atherosclerosis will be for type 2A. However, most common is type 2B. Most common it is type 2B, which is the combined hyperlipidemia, where both LDL and VLDL are elevated. In addition, we have some other types, the dis-beta lipoproteinemia, the hypertriglyceridemia, and the mixed hyperlipidemia, known as the type 3, 4, and 5 respectively. They are comparatively less frequently asked. Mostly you will get the questions on type 1, type 2A, and type 2B. Coming to question number 3, which group of enzyme does not belong to the ribozyme class? Now this is a slightly complicated question. First of all, the answer is poly-A polymerase. Poly-A polymerase is the enzyme which is not a ribozyme. The others have a ribozyme function. So first, what is a ribozyme? What do you mean when you say a ribozyme? The ribozyme is an RNA with catalytic activity. An RNA with catalytic activity. So any RNA with catalytic activity will be ribozyme. Mostly the biocatalysts are proteins, but there is an exception. There is an exception where RNA is acting as ribozyme. The 3 well-known ribozyme which are present physiologically and which you must know about is, first of all, the peptidyl transferase. First is the peptidyl transferase. Second, we have the small nuclear RNA. Second is small nuclear RNA, commonly known as the snRNA. Commonly known as the snRNA. And third, we have the introns themselves. See, the small nuclear RNA is responsible for splicing, and the intron has self-splicing activity. Intron has the self-splicing activity. In addition, the peptidyl transferase in prokaryotes and in eukaryotes, the peptidyl transferase in the prokaryotes and in eukaryotes are different molecules. In the prokaryotes, it would be the 23S ribosomal RNA, whereas in the eukaryotes, it is the 28S ribosomal RNA. In eukaryotes, it will be the 28S ribosomal RNA. So these are the three main ribozymes which you must know about, which are physiologically present. Besides this, large number of ribozymes are being discovered in lower forms. In addition, large number of ribozymes are also synthesized synthetically in the lab to understand the interaction of the various RNA and the proteins. So these are the three physiological ribozymes which are important, which you should know about. Question number 4. Which of the following does not provide a cytosolic NADPH? Here you have to remember two things. In the cytoplasm, you can have both NADH and NADPH. Right? We can have both NADH and NADPH. The NADH will enter via shuttles into mitochondria. It will enter via shuttles into the mitochondria, then it goes to the electron transport chain where it is able to generate the ATP. But NADH will stay in the cytoplasm only. NADH will stay in the cytoplasm only. It performs two important roles. Number 1, biosynthesis. It will provide reducing equivalent for biosynthetic reactions where you are making the fatty acid or you are making the cholesterol. Whatever you are making, there we need the NADPH. And second, it has a very important antioxidant function. So these are the two primary functions of NADPH, both occurring in the cytoplasm. The NADPH does not enter the mitochondria. It cannot give rise to the ATP via the electron transport chain. Its only role is either in the biosynthesis or as an antioxidant. So let's see from where this NADPH is coming. The main source for NADPH is the HMP shunt. I'm sure you know about the HMP shunt. HMP shunt is the hexose monophosphate shunt, also known as the pentose phosphate pathway, also known as the pentose phosphate pathway. Now this HMP shunt gives us two very important components. First, we already said, is the NADPH, and second is the pentose sugar, which is ribose 5-phosphate. That is the ribose 5-phosphate. The ribose 5-phosphate will be used for purine and pyrimidine synthesis. Purine and pyrimidine synthesis. In the HMP shunt, the NADPH comes from two enzymes. The first enzyme is the well-known G6PD, that is the glucose 6-phosphate dehydrogenase. The first enzyme is the glucose 6-phosphate dehydrogenase, and the second enzyme is 6-phosphogluconate dehydrogenase. Most of the NADPH inside the cell in the cytoplasm will come from these two enzymes, which are part of the HMP shunt, but a very small percentage, about 5 to 10% of the cytoplasmic NADPH will come from cytosolic malic enzyme and the cytosolic isocitrate dehydrogenase. These are the two additional sources of NADPH inside the cytoplasm. Inside the cytoplasm, these are two minor sources of NADPH, and they can also contribute small amount to the NADPH production. So that means malic enzyme, it is a source. Glucose phosphate, it is a source. The cytosolic isocitrate dehydrogenase is also the source. So we are left with the ATP citrate lyase. The ATP citrate lyase is not a source of the cytosolic NADPH. It is not a source of the cytosolic NADPH. Question number 5. Which amino acid is responsible for synthesis of urea, creatine and nitric oxide? We have various options: the arginine, the glycine, alanine and methionine. Let's look at the substrates which help in the synthesis of these molecules. The urea coming from ammonia, carbon dioxide and aspartate. The ammonia will give us 1 nitrogen, the carbon dioxide will give us 1 carbon, the aspartate also gives us 1 nitrogen. Similarly, creatine, the donors are glycine, arginine and methionine. And finally, for nitric oxide, we have only arginine. So going by this list, none of the options appears to be correct. But what happens in the urea synthesis? In the urea synthesis, the last step where the urea is formed involves the cleavage of arginine by the enzyme arginase, which cleaves the arginine to give urea and ornithine. So you can say the urea is coming directly from arginine. Urea is coming directly from arginine. Keeping this in mind, urea, creatine and nitric oxide, all of them come from arginine. All of them come from arginine. So the best possible answer in this case would be arginine. The best possible answer in the given list for the amino acid which generates the 3 molecules is arginine. Question number 6, again on ammonia. Ammonia is toxic because it leads to depletion of which substrate of Krebs cycle? Now this is a very frequently asked question. First I will show you the reaction in which the ammonia is getting used up, and it will also show the intermediate or the substrate of Krebs cycle which is there. The phenomena is the reverse of oxidative deamination of glutamate. See, normally what happens, the glutamate is formed by transamination. By transamination we get the glutamate, and this glutamate undergoes what is known as the oxidative deamination. It undergoes what is known as oxidative deamination to release the ammonia in the free form. So in this reaction what happens, the ammonia and the alpha ketoglutarate are formed. The ammonia and the alpha ketoglutarate are formed. But this reaction is freely reversible. As a result, when the ammonia concentration increases, the backward reaction becomes more favorable, because it is a freely reversible reaction. It will depend on the concentration of the substrate at one end and the product at the other end. So what happens, when the ammonia concentration will rise, the backward reaction will become more favorable and the ammonia starts getting chelated. So the backward reaction will also use up the alpha ketoglutarate. So when the reaction goes in the backward direction, the alpha ketoglutarate level will start falling down. The alpha ketoglutarate, you know, is an intermediate in the Krebs cycle or the TCA cycle. If one of the intermediates of the Krebs cycle is decreased in amount, the whole of the Krebs cycle will stop or it will slow down. Main action of the Krebs cycle is to generate ATP. When the Krebs cycle is blocked or it is slowed down, the ATP production will fall. Because the ATP production will fall, those tissues which use more of ATP will be affected first, and that is the brain. So the ammonia toxicity is due to depletion of alpha ketoglutarate. The first organ to be affected will be the CNS. Commonly it presents with encephalopathy. If untreated, can progress to coma. Encephalopathy, if untreated, can progress to coma. The main thing to remember is the oxidative deamination is a freely reversible reaction. When ammonia levels rise, the backward reaction becomes more favorable and alpha ketoglutarate gets depleted. Alpha ketoglutarate gets depleted. So coming back to the question, the answer would be alpha ketoglutarate. Alpha ketoglutarate. Now we have one more question from urea cycle. The glutamate level, it can even be glutamine, any of the two names can be there in the question. Glutamate or glutamine level is increased in blood, urine and CSF due to deficiency of, now this is the characteristic of hyperammonemia type 1. It may be seen in the other hyperammonemias, but most prominently it is seen in the hyperammonemia type 1. The elevated amount of glutamate and glutamine is maximum in hyperammonemia type 1. The hyperammonemia type 1 is due to deficiency of the CPS1. The OTC, that is ornithine transcarbamylase. The ornithine transcarbamylase will cause the hyperammonemia type 2. It will cause the hyperammonemia type 2. The CPS1, for those who don't know it, stands for carbamyl phosphate synthetase 1. It is the carbamyl phosphate synthetase 1. The Isocitrate synthetase deficiency will cause citrullinemia, again type 1, and the arginase deficiency will present as hyperargininemia. Will present as hyperargininemia. So the best answer is CPS1, where glutamate and glutamine level is elevated in urine, CSF as well as blood. In urine, CSF as well as blood. Now question number 8 talks about an alcoholic malnourished patient with respiratory distress. Pulse is very fast. Patient has edema, hypertension and a systolic murmur along the left sternal edge, indicating heart failure. This is a characteristic finding of heart failure. Again, bilateral crepitations were felt in the lungs, again present in the congestive heart failure, and a diagnosis of congestive high output cardiac failure was made. Now this high output cardiac failure is a characteristic term which is used for wet beriberi. It is a characteristic feature of the condition called wet beriberi, which is due to deficiency of vitamin B1. The wet beriberi is due to deficiency of vitamin B1. The wet beriberi is one of the very frequently asked clinical cases in your exam. You should know about it. Let's take a look at the different types of conditions related to the vitamin B1 deficiency. First we have the dry beriberi. Now this dry beriberi involves the CNS, the central nervous system. The most important finding is the altered sensation known as paresthesia. The altered sensation known as paresthesia. In addition, we find the general weakness, atrophy, and the foot and the wrist drop. Another characteristic finding are the drops. The foot and the wrist drop would be another important finding in beriberi. That's a dry beriberi. Then we have what is known as the wet beriberi. The dry beriberi involves the central nervous system. The wet beriberi involves the cardiovascular system. It involves the cardiovascular system, and the most characteristic finding is high output cardiac failure. The most characteristic finding is the high output cardiac failure. It involves the cardiovascular system. Other findings include palpitation, the tachycardia, the aortic regurgitation and edema. Edema is there because of the heart failure. In addition to the dry and wet beriberi, these two are very well known. Sometimes in the children, in the newborn, particularly in the infant, we can find what is known as the acute or infantile beriberi. Now this will be seen only in those conditions where the mother is having chronic deficiency where the mother is deficient, because the child is only getting the breast milk. So when the mother is deficient in B1, again commonly occurs in chronic alcoholics. In those conditions, still the child is being exclusively breastfed. He or she may suffer from the infantile beriberi. As soon as you start giving the top feed, it will contain some amount of B1, and this presentation of beriberi will disappear in the infant. So one disorder due to deficiency of vitamin B1 is the beriberi. The second disorder is the Wernicke's encephalopathy. In Wernicke's encephalopathy, what we see is global confusion and ophthalmoplegia. Global confusion and ophthalmoplegia. In addition, in some patients there can be associated psychosis. When the psychosis is present, the condition is known as Wernicke-Korsakoff psychosis. The Wernicke-Korsakoff psychosis, the term used when along with Wernicke's we have the psychosis. Lastly, please remember, in these patients the loss of consciousness is an emergency. You must immediately infuse 1 dose of thiamine. So the other two main conditions associated with thiamine deficiency, first is the beriberi and second is the Wernicke's encephalopathy, which may present with psychosis also. Now question number 9 talks about techniques. It talks about techniques, and it says in which technique the labeled or tagging antibody is not used. Now we know in ELISA the antibody is tagged with enzyme. It is tagged with enzyme. Same is true for ELISpot. It is a variant of ELISA, which is used to identify the cytokine secretion from the cultures. And then in immunofluorescence, the antibody is tagged with fluorescence. It is tagged with fluorescence, whereas in heme agglutination test there is no tagging of the antibody. In the heme agglutination test there is no tagging of the antibody. Question number 10. Which type of collagen is found maximally in the skin? Now this is a very tricky question, because we have certain tables where we memorize which particular collagen is there in which of the tissues, but remember, collagen type 1 is most predominant in all tissues. Everything else which is there is on the background of the collagen type 1, because collagen type 1 will provide the strength to the extracellular matrix in all the tissues. It is the most ubiquitous type of collagen. So even if in the skin the type 3 is present at an amount of about 20%, the remaining 80% still happens to be the collagen type 1. Collagen type 1 is still the predominant form in the skin also. So which type of collagen is maximally found in the skin? The answer remains collagen type 1. I'm sure you know collagen type 1 has the formula glycine XY, repeating n number of times, where a large amount of X is proline and the large amount of Y can be hydroxyproline. Because of this characteristic structure, the collagen type 1 forms a very tight helix. The tight helix has high strength. It has a very high amount of strength. Unlike the alpha helix is somewhat open, it is not so strong, whereas the tight helix is very, very strong. So collagen type 1 is maximally present in not only skin but most of the tissues. Next we come to the MRP2 deficiency. The MRP2 deficiency. What is MRP? Let's take a look at MRP. It is a part, as you can see from the example, it is related to the jaundice. So we'll see what happens from where the bilirubin is come and what is the pathway. So you can immediately decide where the MRP2 is there. The heme is first converted to biliverdin. The biliverdin is then converted reversibly to bilirubin. This bilirubin is unconjugated and it is transported with the help of albumin to liver. At liver, there is facilitative transport. And inside the liver, the bilirubin undergoes conjugation. The conjugation occurs with the help of glucuronic acid. Conjugation occurs with the help of glucuronic acid, giving us the bilirubin diglucuronide. The enzyme catalyzing this reaction, the conjugation, is the BIL-UGT. The UGT stands for UDP glucuronyl transferase. The full name, if you want, is bilirubin UDP glucuronyl transferase. Bilirubin UDP glucuronyl transferase. This bilirubin diglucuronide is then secreted into the bile canaliculi. The transporter protein is MRP2, meaning the MRP2 is transporting the conjugated bilirubin. The MRP2 is transporting the conjugated bilirubin. Just for the sake of record, I will quickly complete this. The bile canaliculi will release the bilirubin into the CBD. From CBD it will go to the second part of duodenum. From second part of duodenum it goes to the rest of the small intestine, from where it goes to the large intestine. In the large intestine it is acted upon by the gut flora. As a result of the action of gut flora, three things happen. Deconjugation, the conversion to bilirubin, the conversion to urobilinogen, and the conversion to stercobilinogen. After this, they are excreted in the feces. The stercobilinogen, on getting exposed to oxygen, forms the brown pigment which is responsible for the color of the stool. It forms the brown pigment which is responsible for the color of the stool. Knowing this, let us look at the question. The MRP2 deficiency will be seen when there is elevated conjugated bilirubin, which is seen only in the Dubin-Johnson syndrome. In these three conditions, the unconjugated bilirubin is elevated. In these three conditions, the unconjugated bilirubin is elevated. Going to question number 12. Severe jaundice is seen in. Now, in Dubin-Johnson, the jaundice is mild. Again, we know that it is due to defect of MRP2 deficiency. Whereas in Crigler-Najjar syndrome, the activity of the enzyme BIL-UGT is nil. In type 2, the activity is less than 5%. In Gilbert's, the activity is less than 30%. So obviously the most severe type of jaundice is likely to be seen in the Crigler-Najjar syndrome, because the conjugation is not occurring at all. All the bilirubin which is being formed, will stay in the circulation. So we have the severe jaundice in Crigler-Najjar syndrome type 1. Whereas in type 2 and Gilbert's, because some residual enzymatic activity is there, we can stimulate this activation by the drug called phenobarbital. The phenobarbital will stimulate the activity of all the residual enzyme, and the bilirubin which is getting accumulated will be less. The type of jaundice will be less severe. Then we have the condition called Menkes disease. Menkes disease is an inherited collagen defect. It is an inherited collagen defect. Now what happens in extracellular matrix? In extracellular matrix, lysine of collagen is oxidized. The enzyme catalyzing the reaction is lysyl oxidase. The lysyl oxidase requires copper as a cofactor, which is transported into the extracellular matrix by ATP7B. ATP7B will transport the copper into extracellular matrix, makes it available for the lysyl oxidase, which will oxidize the lysine. The ATP7B is deficient in the condition known as the Menkes disease. Because ATP7B is not there, the copper will not be there. The copper is not there, so the lysyl oxidase cannot work. The lysyl oxidase is the enzyme which will not be able to work. The lysyl hydroxylase and prolyl hydroxylase both require vitamin C for obviously hydroxylation of the respective amino acids, meaning lysine and proline. They can oxidize, they can add the hydroxyl group to the lysine and proline, converting it to the hydroxylysine and the hydroxyproline. Now next question is about the dietary fiber. Which of the following is not a dietary fiber? The options are cellulose, gum, pectin, inulin. Now interestingly, all of them are dietary fibers. Let's take a brief look at dietary fibers. What do you mean by dietary fibers? Basically, dietary fibers are polysaccharides. They are polysaccharides which are not digested, Some of the important dietary fibers are cellulose, inulin. The cellulose is made up of only glucose, the inulin is made up of only fructose, meaning these are the homopolysaccharides. So among the homopolysaccharides, the cellulose and inulin are dietary fiber. In addition, commonly in our diet we also find lignin and pectin. Lignin and pectin would be what are known as the heteropolysaccharides. The building blocks are not all the same. All the building blocks are not the same, so they are called the heteropolysaccharides. They are commonly present in a large amount of food. Another type is gum, commonly present in the raisins. Normally it is not a part of our diet. So going back to the previous question, the cellulose, the pectin and inulin are commonly present in the diet. The gum, in spite of being the dietary fiber, is not commonly present, so it would be the preferred answer. Strictly speaking, gum is also not a dietary fiber. Gum is also a dietary fiber. Strictly speaking, gum is also a dietary fiber. However, it is normally not present in the diet. Then we come to the formation of the catecholamine. Norepinephrine to epinephrine requires which amino acid? Now let's take a look at the formation of the catecholamine. The picture will become very clear. The catecholamines require large number of the coenzymes. First, the conversion of tyrosine to dopa requires tetrahydrobiopterin. Conversion of tyrosine to dopa requires tetrahydrobiopterin. The conversion of dopa to dopamine requires PLP. Basically, it is the active form of vitamin B6. Active form of vitamin B6. Conversion of dopamine to norepinephrine. Dopamine, which happens to be the first catecholamine, can be converted to norepinephrine using copper and vitamin C. These two things are also required for the collagen synthesis. We just seen they are also required for collagen synthesis. Finally, we come to the conversion of norepinephrine to epinephrine by the enzyme PNMT. Here you can see the S-adenosylmethionine is acting as the carbon donor. Basically, it donates a methyl group to convert the norepinephrine to epinephrine. It converts the norepinephrine to epinephrine. So, conversion of norepinephrine to epinephrine requires methionine in the form of S-adenosylmethionine. It requires methionine in the form of S-adenosylmethionine. Then we come to the mitochondrial DNA. True regarding mitochondrial DNAs. The question on mitochondrial DNA nowadays is coming again and again and again. This is one question where you should know everything. The mitochondrial DNA is a very small topic, so we'll quickly take a look at the mitochondrial DNA and come back to this question. The mitochondrial DNA, we are talking about the mitochondrial DNA. Mitochondrial DNA is double stranded. Just like nuclear DNA, this is also double stranded. However, unlike nuclear DNA, this DNA is circular. In addition, the mitochondrial DNA is small, barely 16,000 base pairs, 16569 base pairs, barely 16,000 base pairs. These 16,000 base pairs code for 13 proteins, 2 ribosomal RNA and 22 transfer RNA, meaning a total of 37 genes. A total of 37 genes, 13 coding for protein, 2 coding for ribosomal RNA, remaining 22 code for the transfer RNA. These 13 proteins all belong to electron transport chain. The electron transport chain itself has a total of 67 protein subunits, out of which only 13 come from the mitochondrial DNA. Besides being small, the mitochondrial DNA does not have introns. There are no introns. Result of no introns: splicing not required. First very important thing, the splicing is not required. And second thing, because splicing is not required and the transcription, replication, translation occurs all in the same compartment, simultaneous transcription and translation, simultaneous transcription and translation occurs. The transcription is not yet complete and already the translation has started occurring. So simultaneous transcription and translation occurs. Besides having no introns, they have very few untranslated sequences. They have very few untranslated sequences. However, one very unfortunate finding for the mitochondrial DNA is it has the high mutation rate. It has the high mutation rate, typically 5 to 10 times higher, typically 5 to 10 times higher. And lastly, there is variation in translation of genetic code. There is variation in translation of the genetic code. Let's take a look at the variation in the translation of the genetic code. In the first column, I'll write the codons, then I'll write the normal translation, and finally we'll mention the mitochondrial translation. Finally, we'll mention the mitochondrial translation. The codon, the normal translation, and the mitochondrial translation. Starting with the codon called UGA. Everyone knows about the codon UGA. It normally codes for stop codon. It normally codes for stop codon. But within the mitochondria, the UGA will code for tryptophan. The UGA will code for tryptophan, right? The second one is AGA and AGG. Normally these two code for arginine. AGA and AGG normally code for arginine, but inside the mitochondria they work as stop codon. Inside the mitochondria they work as stop codon. Then we have the codon AUG. In eukaryotes, this is the only codon for methionine. So in eukaryotes, you find methionine attached at any codon, it is going to be AUG. But in prokaryotes, the AUG gets translated to formyl methionine. It gets translated to formyl methionine. And last, because we have used up the codon for methionine, we need another codon for methionine, which happens to be AUA. Normally AUA will code for isoleucine, but in mitochondria it codes for methionine. So these are some exceptions to genetic code which you should know when we come about the mitochondrial DNA. When we are talking about mitochondrial DNA, these are some additional points related to the translation which you should know about. Okay, so that was about the mitochondrial DNA, details about the size and details in the difference for a translation. Protein not synthesized in liver. Now this is a straightforward question. Albumin is synthesized in the liver. The plasma enzymes are synthesized in the liver, the ALT and AST. The accused phase protein is also synthesized in the liver. The immunoglobulins will come from the plasma cells. The immunoglobulins will come from the plasma cells. These are the mature B cells which have started producing the immunoglobulin. Now substrate linking Krebs cycle with urea cycle. For this we will take a quick look at a part of the Krebs cycle and then come back to this question. So what happens in one of the reactions, the citrulline combines with aspartate. Citrulline combines with aspartate in the presence of the enzyme, called arginosuccinate synthetase. The arginosuccinate synthetase will combine citrulline and aspartate to give us, as the name suggests from the enzyme, arginosuccinate. The arginosuccinate is then quickly cleaved into two. It is then quickly cleaved into two. The first being arginine, and the second being fumarate. The second being fumarate, the reaction is catalyzed by arginosuccinase, also known as the arginosuccinate lyase. Both terms are used, either you call it arginosuccinase or you call it the arginosuccinate lyase. Now the fumarate which is formed enters the mitochondria. It enters the mitochondria. In the mitochondria, the fumarate is first converted to malate. The malate is then converted into oxaloacetate. The oxaloacetate is transaminated to give us aspartate. Remember, all this is occurring inside the mitochondria. The aspartate is then transported outside by the glutamate aspartate shuttle, also known as the citrin. The name of the transporter is citrin. It is the glutamate aspartate transporter. Now the question can be asked in two ways. If they mention substrate, obviously the most important name would be the fumarate, the bridge between the urea cycle, and these are the reactions of the TCA cycle. These are the reactions of the TCA cycle. They are the reactions of TCA cycle. So the bridge between the urea cycle and TCA cycle is the fumarate. However, sometimes they can ask you about amino acid. So remember, the fumarate is not an amino acid. In that case, the answer will become aspartate. If they ask about amino acid, the answer is aspartate. If they ask about the substrate, obviously the answer should be fumarate. We will only mark aspartate when they mention amino acid, otherwise the preferred answer will always be a fumarate. The fumarate is the preferred answer as a bridge between urea cycle and the TCA cycle or the Krebs cycle, as a bridge between urea cycle and the TCA cycle or the Krebs cycle. The answer when it says substrate should be fumarate. If it mentions amino acid, in that case the answer would be aspartate. If it mentions amino acid, then the answer would become the aspartate. Then you go to question number 19. In type 1 diabetes mellitus, which of the following is true? When we say type 1 diabetes mellitus or any diabetes mellitus, we are actually talking about a fasting, or you can say the starvation state. We are talking about fasting or the starvation state. Now there are four types of question, which all of them will be approached in the same manner. Let's take a look at which are those questions and then come back to this question. First, whether you are talking about fed state or the fasting state. That's scenario 1. Scenario 2, whether you are talking about the enzyme active in the phosphorylated or the dephosphorylated form, in which form the enzyme is active. Third, are you talking about the insulin glucagon ratio? The third is insulin glucagon ratio. And the fourth is obviously the actions of insulin itself. Actions of insulin directly. Actions of insulin directly. Now let's start one by one. First, talking about the fed and the fasting state. So in the fed state, what will happen? There will be increased glucose. When the glucose increases, there will be increased insulin. So when I say fed state, I am basically asking about enzymes activated by insulin. When I say fasting, I am talking about enzymes activated by the counterpart, that is the glucagon. Now coming from insulin, most of the enzymes by insulin are dephosphorylated. Most enzymes undergo dephosphorylation. So what this means, if the insulin is activating some of the pathways, the enzyme is active in the dephosphorylated form. If the insulin is downregulating some of the pathways, the enzyme in the dephosphorylated form is inactive. In the phosphorylated form, it will be active. So again, we need to know the pathways which are activated by insulin and the pathways downregulated by insulin. In activated pathways, enzyme is active in the dephosphorylated form, what the insulin is causing as a change. Whereas in the inactivated pathways, the enzyme will be working in the phosphorylated form. Okay. Next we go to the insulin glucagon ratio. This is very, very interesting. Please watch very carefully. The insulin glucagon ratio. Now first, you have to understand that insulin and glucagon have a reciprocal relationship. If insulin increases, the glucagon will decrease. If insulin decreases, the glucagon will rise. Now let's say that insulin level increases. We are talking about the fed state. In that case, the glucagon will become less in amount. The numerator is increasing, the denominator is decreasing, the overall value increases. The overall value increases. Contrarily, if insulin decreases, the glucagon will increase, reciprocal relationship. So what is happening? Numerator is decreasing, the denominator is increasing, the overall value becomes less. So just to simplify, see what has happened here. The increase in insulin causes increase in the ratio. A decrease in insulin causes a decrease in the ratio. So whenever we are talking about the insulin glucagon ratio, we don't actually need the term glucagon ratio. The trend of the ratio will be the same as the trend of insulin. If the ratio decreases, that means the insulin has decreased. If the ratio increases, that means insulin has increased. So again, insulin glucagon ratio will have the similar effect as that of insulin, because that follows the change of the insulin. So ultimately we are coming to what are the actions of insulin. Now there are multiple pathways which are activated by insulin, multiple pathways which are down-regulated by insulin. But it's very difficult to memorize which is activated, which is down-regulated. We have so many pathways. How to remember? A very simple way to remember the actions of insulin. See, when is insulin increasing in amount? The increase in insulin occurs when there is increase in glucose. Reason? The insulin is supposed to decrease the concentration of glucose. The insulin has been produced in response to increased glucose so that that glucose can be consumed. This means insulin will stimulate all those pathways which are going to consume the glucose. Alright, that's point number one. Insulin activates the pathways which consume glucose. Now insulin is the preferred fuel. So as long as glucose is the preferred fuel. So as long as glucose is there, insulin will downregulate all those pathways which produce fuel. Fuel means glucose, fatty acid, ketone bodies. This means insulin increases glucose consumption and it decreases metabolic fuel production. it decreases metabolic fuel production. So keeping this in mind, the list becomes very simple. The list becomes very simple. Let's see the pathways where the glucose consumption is targeted. Glycolysis, glycogenesis, HMP shunt. HMP shunt will also make use of glucose. Now when a lot of glucose is there, it will be converted to pyruvate, it will go to the Krebs cycle. In the Krebs cycle we will get the acetyl coenzyme A. The acetyl coenzyme A can be directed towards lipogenesis, or it can be directed towards cholesterol synthesis. Lastly, when we have excess of glucose, a lot of intermediates of the Krebs cycle is there. Some of them can be diverted towards the amino acid synthesis. So these are the pathways which are upregulated by insulin because they will consume the glucose. The pathways which produce alternate fuel, they will be down-regulated. Remember, those pathways will be down-regulated. They include first production of glucose, they are glycogenolysis, and second gluconeogenesis. These two directly produce the glucose. Then we have the lipolysis. As a result of lipolysis, more and more of fatty acid will be formed, and finally we have the process known as ketogenesis. What this means, the insulin will activate all these pathways. The insulin will inhibit or downregulate all these pathways. In all these pathways, the enzyme is active. The enzyme is active in dephosphorylated form. In dephosphorylated form. Just a sec. Enzyme active in dephosphorylated form. Whereas all the enzymes of this pathway, enzymes active in phosphorylated form. Alright. So this will help you to remember all these four types of question. Let's come back to the question. In type 1 diabetes mellitus, we said it is a fasting state. Fasting state means the effect of insulin will be decreased. Either insulin is not there or its effect is not there. Now question asks, which of the following is true? If insulin is not there, lipolysis should be increased because normally it is inhibited by insulin. The protein catabolism will be increased to provide for gluconeogenesis. The glucose output will be increased again via gluconeogenesis, and the glucose uptake is decreased because insulin action is not there. Glucose uptake by the cells is decreased. So the best answer is increased lipolysis. In fasting state, we mostly find the increased lipolysis. Coming to the last question. the Alzheimer's disease is associated with increase in which of the following apolipoproteins proteins. So this is a recent discovery. A lot of papers have shown that the Alzheimer's disease is increasingly showed having strong association with ApoE4. So these are the questions which have been asked in the NEET PG 2019 exam. Please go through them. Some of them are likely to land up in your exam again. Most of them are very important topics which we repeatedly discuss in our class. In the next lecture, I will come up with the AIIMS May questions which were asked, so that you can go through them, and if you have any doubts, you can send them to me. Thank you very much. Thank you very much, and please keep revising. Watch the videos, and if you have any queries, please get back to us.